Welcome, everyone. Today, we have a very, very special person, Laura Santambrogio. She's from Italy. She has a little accent from Europe, like me. She's an MD and PhD, and she received her PhD from Padua University, one of the first university in Europe with Bologna. And I have been there. I saw the beautiful anatomical theater. I think it was the first one in the world. It's absolutely beautiful. And then she went as a postdoctoral fellow in NYU and Harvard Medical School. And today she moved to Weill Cornell, and she's associate director of precision immunology. And I tell you, she has a lot of precise knowledge on immunology and the lymphatic system. And she's also professor of radiation oncology, physiology, and biophysics. Thank you, Laura, for being with us.
Thank you. Thank you for having me.
It's a great honor to talk with you. I think it's going to be very, very interesting. Laura, let's maybe start with the basics. And I know you have some slides about the main functions of the lymphatic system.
It's really a pleasure to be here today, and I'm obviously always very happy to talk about the lymph and the lymphatics. It's a research that is very close to my heart. We've been working for many years, and the lymphatic system is often described as the third circulation, as compared to the arterial and venous system. So there are three major functions of the lymphatic system. One is fluid homeostasis. The second is tissue homeostasis. And the third one is immunosurveillance. Even though I describe here as being three functions, but they are all connected with each other. So what is fluid homeostasis? We know that there are around, in the human body, around 8 to 10 liters of interstitial fluid that are formed every day. And they derive from the oncotic pressure inside the arterial end of the capillaries bed, which push out all the nutrients and all the molecules that they need to go into an organs to be picked up as nutrients. And so every time you have a movement of protein, lipids, carbohydrate molecule in general into the interstitial fluid, there is water that comes with it. And it was thought, and this is the so-called Stirling principle, that the majority of the interstitial fluid would go back to the venule end of the capillaries and go back to the circulation. However, during the last 20 years, several labs have shown using fluorescence tracer, molecular tracer, that actually almost 90% of the interstitial fluid does not go back to the venule end, but enter the lymphatic circulation, the lymphatic capillaries. There is only one exception to this rule, are the mesenteric lymphatics, because in that case, the absorption does not go only to the lymph, probably a minority to the lymph. The majority is to the portal vein that goes to the liver. But besides this exception, in all other systems, the majority of the interstitial fluid, over 90%, goes into the lymphatic capillaries and then eventually to the circulation.
I prepare that because I know it's very important. In 2010, Levick and Mitchell, they published in Cardiovascular Research recapitulation about the new concept of a Starling, and they really show that if you want, this is the old model. You have a filtration and reabsorption by the venous system. But when they redo their calculation and added the glycocalyx at the coat outside of the cell, they realized there's basically almost no reabsorption in the vein. And when I was in school, I had like 10% reabsorbed in the lymphatic, 90% in the vein. Then they went the other way, 90% in the lymphatics, 10% in the vein. But I think they went to 99% almost. Now they say there's almost nothing.
you have a lot of waste product there, and you don't want to bring them back to the general circulation, because you may compromise the equilibrium of oxygen transport. At the same time, though, these are a treasure trove. When people say it's something that is junk to you, but can be a treasure for me, right? And that's what it is for a lymph node. The waste product for a lymph node are information on what's going on in that organ. And that's why I think this is a concept that has not been explored enough.
The artery is okay to deliver some substance, protein, lipids, etc., to the extracellular fluid, but the vein doesn't recover anything. All those, whatever it is, coming from the extracellular fluid, it will modify the pH too much. So the lymph is doing that job. So really,
like protein, right? And a pathogen, for example, as a physician, that if you have septicemia, if you have a pathogen in the blood, it's extremely complicated to control. Even very high dose of antibiotics does not work. And whereas in the lymphatic system, that's the normal way. They go to the lymph node and you have all these cells which are ready to kill the pathogen. That's really why you want them in the lymphatic system. So it's a combination of all these things. Protein concentration, you cannot change. You go in hyperosmolarity if you dump all this amount of protein into the blood. In the lymph, you have around 30 milligram of protein per ml as compared to the blood, which is almost double. So you don't want this extra amount of protein. And if there are, again, change in pH, like during inflammation, during the cancer tissue microenvironment, you don't want all these ions to go into the blood. They would change, the electrolyte balance and so on. Whereas the lymphatic system can manage them. You can still maintain body homeostasis, even though you are collecting all this lymph waste. Why do we need the drainage into the lymphatic and not the venous system? And probably, even though there is not a lot of research on it, probably the major reason is that the lymph is very important for the maintenance of organ homeostasis. Like in the blood, protein concentration, electrolytes, lipids in general, pH, they have a very narrow range of change. After that, you really compromise body homeostasis. If you look at the pH, it can only go, normally the pH is 7.4, it can only go down to 736 or 744. These are the range of pH. Above and below, it's not in homeostasis. And the same for electrolytes. They have a very narrow range of concentration. And so every time you change this concentration, you can compromise oxygen transport, which is the major role of the blood. And that's why you need a system to drain all the waste without compromising the blood circulation and homeostasis. Because in many situations, during inflammation, during trauma, all these parameters change. The pH may change. We know that during inflammation, the interstitial pH can go down to 5.5. Protein concentration can change. And so you need a system that is capable of draining all these changes from parenchymal organ without compromising the homeostasis.
So in the center, this is called the Bohr effect. We can see on the blue line at 7.2, we release oxygen. We have a lot of CO2.
Yes.
Go below that, it's going to completely change viability of an organism. So
that's right. Yes.
Go there. So it's so important, and very few people talk about that. It's so important to understand.
I know. That's why, like, we know during inflammation or during tumors, in tumor microenvironment, sometimes the pH, there is hypoxia, and sometimes the pH goes down to 5.5. So if you start draining all this fluid into the blood, you would compromise a lot of these parameters that they need to stay in this physiological range.
At pH 5, we're probably not alive.
Right, not in the blood. In the lymph, at least at the beginning when the lymphatic fluid enters the interstitial fluid. The same, for example, for, let's say, potassium. We know that increase or decrease in potassium can give arrhythmias because you need a certain concentration of potassium for the heart, to pump, and so on. Or protein concentration. You can have in diabetes when, for example, the glucose is very high, the protein, the lipids, are very high. You can have hyperosmosis.
Do we know the window for the lymph? How much?
We don't. We don't, and actually would be very important because, as you can see in this next slide, you understand what is the huge range of changes that this organ, lymph, is capable of withstanding without compromising homeostasis. And so I think it would be extremely important to understand how much protein concentration can change, or pH, or again, electrolytes, and so on. You can see during a trauma, you can see how much red blood cells are in the lymph, right? Just by looking at the color. Or during an infection, these two are infections, and this is the normal lymph. Or even after a meal, just by looking at the yellow color, you can see how much more lipids you have in the lymph as compared to...
Laura, this is a mesentery, right? Because the lymph is not white.
Yeah.
Mesenteric, the lymphatic of mesenteric lymphatic of the abdomen. I was talking with
Oh, right. Yes. Yeah.
And you told me that the lymph often have a little bit of red corpuscle that reverse a little bit into the lymph, and especially when you do exercise. He said after exercise, the lymph is not transparent and not like here, but really clear, transparent, like the white of an egg. He was saying it's pink. Is that something you have seen, or?
Well, we never look after exercise. However, every time you have a breakdown, for example, you can have a little trauma during exercise. And so whatever happens in the muscle, which is drained by the lymph, that's why you have, like, if you have few red blood cells, they would give a different color to the lymph. That I think that the point to bring home that is really important is how this fluid is really sensitive to whatever is going on in any particular organ, because it drains that, each parenchymal organ. And the blood is not like, if you have an infection, you don't see all these changes in the blood, but you see them in the lymph. And so that's why, the maintenance of organ homeostasis is certainly an important function of the lymph. And this goes together with the immunosurveillance, because all this information that comes from every parenchymal organs are transported to the lymph node, which is where you want them, because is a conduit to tell the lymph node what is going on in these organs, because you would receive immune cells, apoptotic cells, exosome, vesicle, apoptotic body, protein, peptide, lipids, metabolites from each organ. It goes to the lymph node. And so it really informs the lymph node if there is an infection in this organ, or if there is any disbalance in whatever, physiology of the organs. And so you basically can put the two things together. You control and maintain organ homeostasis, and at the same time, if the homeostasis is not there, you are informing the lymph node that something it is changing. And the lymph node is the perfect conduit to mount an immune response to whatever, is going on in that organs. And here I just put, this was actually, we were discussing about Anatoly before we went online, and he was a dear friend, and he was a great scientist in the field. And that's why I put a movie that he made, and you can see these are red blood cells. So these are red capillaries, and this is the lymphatic that is pumping the lymph towards the lymph node. And he was able to measure the amplitude, the strength of the pumping, as well as the frequency. And the lymph in this way is carried to the lymph node. And there are pacemakers in the lymphatic that depolarize with calcium, similar to the pacemaker that we have in the heart. And every time there is a depolarization, there is a contraction of the lymphatic to carry the lymph forward.
That's in the human, right?
This was in rat, not in.
Right. So when you see, on the right, you see blood vessels, right, going very fast.
Two, yes, these two. One is here. I don't know if you see my cursor, and one is here. Yeah.
The slow rate of the lymph help antigen antibody contact. It's helping to be a bit slower. If it would be so fast like that, the immunosurveillance role would be more difficult, probably, no?
The rheology is different. There are less immune cells. There are in general less cells in the lymph as compared to the blood. I don't know any study that measure this, antibody protein recognition specifically in the lymph. But is definitely the flow is definitely much slower as compared to the blood. Yes, that's a possibility. Yeah. And so basically the lymph will enter the lymph node, and this is like a cross section of a lymph node. You can see the afferent lymph. This is a human lymph node. The afferent lymph will enter, and they're called subcapsular sinuses, which are like almost like pool in which the lymph will enter. And around these subcapsular sinuses, you have macrophages, dendritic cells, which are specialized immune cells, which can pick up phagocytose, pick up and eat bacteria, any pathogens, and also the self protein that is incoming. And they process it and they present it to the T and B cell. So that's immunosurveillance. Dendritic cells and macrophages are instructing the rest of the immune system what is incoming in the lymph node. And that's why these sinuses are very close to the afferent lymphatic. It's very important. There is an anatomical barrier here. It's around 80 kilodalton. And so big protein and certainly pathogen cannot enter through here. But the fluid, yes, it can enter because the fluid will be reabsorbed to the high endothelial venule. And so this is a way for these like 8-10 liters that I was describing before of interstitial fluid that will be reabsorbed into the blood circulation, but only the fluid. And this is very important, not the pathogen, because as you know, septicemia is extremely difficult to control at the medical level when you have bacteria in the blood. And so that's why there is this barrier, because if the pathogen is not picked up here, you have other possibility. You can pick it up in the medullary sinuses. If you don't pick it up there, it can exit through the efferent lymphatic, go to the next lymph node, and there is a possibility again to be phagocytosed by dendritic cells and macrophages. So basically, the system is geared to make sure pathogens stay inside the lymphatic system. They don't enter the blood. And that's why there are all these anatomical barriers. And the lymph node is extremely efficient. We did this experiment and we did a lot of mathematical modeling, which I'm not presenting today.
I always was wondering, so we have a zone in the cortex here, the B zone, when you have these follicles. And
where is AB, right? Yeah.
So a dendritic cell is a cell that presents antigen. It's called antigen presenting cell, APC. We have the macrophage, lymphocyte T. There are a few of them, and one of them is a dendritic cell. And this dendritic cell is different here. They call it the follicular, and it comes more mesenchyma than the lymphoid aspect. So why do we have different type of dendritic cell in the B lymphocyte? What's different there?
In the node, we have actually many different types of dendritic cells. There are several subtypes. I don't know how, in depth you want me to know.
No, I only know 3, but it's already so.
Oh, okay. Conventional dendritic cells, that's what people used to call myeloid dendritic cells, now they're called conventional dendritic cells. Then you have the plasmacytoid dendritic cells that they make high amount of interferon to fight viral infection. And then you have the follicular, which are, they are like helper for B cells and antibody production. And so all together, the function though of these dendritic cells cells is to capture pathogen, capture, proteins, and to present them to the immune system to process, because as you know, differently from antibody that recognize protein three-dimensionally, the T cells, they recognize pieces of a protein. And so that's the major role of the dendritic cell, to process this protein. Some are self-proteins, some they come from pathogen, and tell the T cell, this is what I see right now. So, if it's a pathogen, you need to mount an immune response towards this pathogen.
Laura, are you saying the dendritic cells, they have dendrite, they have a lot of arms? Those
yes, yeah.
3D, like the T cell doesn't do it. The dendritic get the shape of those antigen foreign substance.
Yes. Many of these, like in this conduit system, the one that I described before, in which there is an 80 kilodalton barrier, the pathogen cannot enter, but smaller protein can. And you have all the dendrites of dendritic cells that enter the conduit, and they're like filtering, basically. They are taking up all this antigen for presentation. And they will, upon presentation, they will help B cell transitioning into more mature B cells and making antibody, plasma cells, making antibodies specific for a particular antigen, and the T cells that are specific, again, for whatever antigen is presented. If it's presenting in a certain way that the immune system thinks that need to be activated, so mostly, pathogens. Yes, that's a major role. I describe the three major component and function of the lymph in the lymphatic system, which is fluid homeostasis, so maintenance of the volume, fluid volume in the body, the tissue homeostasis, so making sure that whatever is going on in any particular parenchymal organ, it then goes through the lymphatic system. So all this fluid, it's carried into the lymphatic system, not in the blood. And then immunosurveillance. And I think, as you know very well, when this system breaks down, when there is the inability of the system to function, we have lymphedema. An additional slide that shows that when the system breaks down and does not work properly, you start seeing, for example, bacteria. Here we injected some bacteria in the foot pad of a rat, and normally they are transported beautifully to the lymph node, and here you can see they start exiting, and they go into the tissue. So the person would, in this case, it's an animal, it's a rodent, but it would develop an infection. And
so with aging, you have hyperpermeability of the collective.
Yes, yeah. And we are studying in the brain. Actually, we have several models in collaboration with Helene Benveniste, Yale, and Joni Kipnis, Washington University. And when we block, and Kari Alitalo in Finland, when we block the lymphatics at the deep cervical node or superficial cervical node, we see that there is an increase in the pressure, intracerebral pressure. It's not permanent, because then the system tries to re-equilibrate itself, and we start seeing metabolomic change, redox imbalance, inflammation. We start seeing an increase in IL-1, for example. And so these are all signs that the system is not at equilibrium and it gears towards an inflammatory.
So if you block the lymphatics, you have oxidative stress change.
Yes.
You found it was a state equivalent to premature aging.
These are all signs that they could eventually bring to a premature aging, yes, because the system, again, is not an equilibrium. We know that increased inflammation, it certainly is a conducive factor for aging, premature aging. And that's what happened, because, if you view the lymphatic system as a way to carry all these waste products away from the organ, if you don't have it because, you know, we are compromising the circulation, this waste product remain in whatever organ we are studying. And they generate an inflammatory signature and a signature of oxidative stress. And neurons are particularly sensitive to oxidative stress. So this could also bring to a neurodegeneration, or at least it could be a conducive factor for a neurodegenerative disease. There are in the literature, you know, several people that look into the Yoni Kipnis Washu, probably is the person who published the most on when you block the lymphatic system, you have a accumulation of tau, accumulation of protein, which are associated with the deposit that you normally see in Alzheimer's. There are studies also, you know, Helene Beniste, Yale, I mean, and other groups that the lymphatic don't function as properly in dementia as compared to a person that does not have dementia. So you have, they increase even further the neurodegenerative process because, again, they do not transport all this waste product away from the brain.
June 2024, you just wrote an article. Basically, what you say is the composition of lymph reflects the anatomical, physiological activity in the region from which it drains. So every part of the body receives the signature from all the organs, all the parenchyma. And it's absolutely amazing if we could, now we use cannula, but if we could easily have a biomarker of disease so much earlier, but unfortunately, it's not so feasible in human, right?
That's what we did in this paper that we published really 2 weeks ago. We collect the lymph from the cervical, the one that drains to the cervical node, and the one that drains to the mesenteric node. And we show how different they are because they come from different organs. You know, in the brain, I mean, it is intuitive, but it was never done before. So like in the one from the brain that drains from the meninge, we found a lot of brain-associated protein, whereas in the mesenteric, we found, you know, even the microbiome. Under it, we collect not only from mice and human healthy, but also people with Crohn's disease or mice with a model, you know, of Crohn's disease. And we show the microbiome that, you know, when you compromise the gut barrier, a lot of these bacteria enter into the lymph and they go to the draining lymph node. And you don't see them in the cervical lymph node. And so it tells you how districtual it is, how, you know, it really drains a certain anatomical region. And the lymph nodes are very good. That's why I put that slides on the filtration ability, because they are extremely good in retaining all this information. We show that only when you really inject a milligram of a protein, you start seeing some traces coming out from the lymph node. If not, these dendritic cells, macrophages, they can handle all this amount of protein that enter, and they can process it. They, you know, can cut them up to present it to T cell.
But the lymph recover from any organ, next to the liver, next to spleen, next to the intestine, all the metabolism, but you have also the from the cytoplasm, the organelle, the ribosome, the mitochondrial Golgi apparatus, from the nucleus, from all that, plus if there's cell death, apoptosis, if there's cancer cells, all that in every region have a different signature. And that's what the cover and not the blood, right?
Yes, yeah. If you take the blood, everything is mixed in. We actually calculate, we did a quantitative analysis. Some of the protein that we analyze are 1,000 times more concentrated, tissue-specific protein that comes from a certain organ are 1,000 times more concentrated in the lymph as compared to the blood, because you are taking it directly from the organ. You know, they are not even entering the so-called afferent lymph, which is the one that goes from the organ to the lymph node, so before it enters the lymph node. The problem is, as you say, biomarker, that would be fantastic. It's just that it's complicated to collect. The blood, you know, it's easier. And Olszewski actually was the one who published a paper with him like 20 years ago on the human lymph in the skin, because they were able to cannulate a lymphatic in the skin, and then as the person was walking around, the lymph would be collected into the tubes, and we did a proteomic analysis of it. But you can do it in the lymph, you can do superficially. You know, now, I mean, there are surgeons that are more amenable to collect some of the lymph, like during surgery, or in Crohn's disease, we were getting specimens from the surgeon. You know, sometimes when there is a strong inflammation and drugs are not working, they need to resect a part of the intestine. And that's what we were getting. And so many times associated with it, there are a lot of lymphatics, and there is also a little piece of a lymph node. And so we were able to cannulate the lymphatic from the intestine to the lymph node. And that's where, you know, we really analyze the content of the lymph. So in the future, you know, if surgeons are more amenable to help, it would be possible maybe in other organs as well.
Yeah. So the lymph, you said, has about 1,000 times to 1,500 times more antigen, self-antigen, basically, than the blood.
Yes, yes.
So let's talk about the role of the lymphatic endothelial cells, LEC, has an active immunological response, because ago it was just a passive cell like any endothelial cells. And I'm going to give you a slide from one of your interview to help you.
So it was thought, you know, that it was basically lymphatic were just a conduit for the lymph to go by, to go to the lymph node.
One second. This is a lymphatic capillary. This will be the parenchyma cells. Those are the dendritic cells. Those are what, dead cells or something like that, or apoptosis. And what are those? Those are lymphocytes, probably?
Lymphocytes, immune cells in general.
So what's amazing, so we enter, this is lymphatic endothelial cells, and we have the cells with that glycocalyx, that cell coating here. And that's super important for presentation of the antigen, right?
The lymphatic endothelial cells, I mean, this is a very hot field right now. What is the role of lymphatic endothelial cells in antigen presentation?
So basically that glycocalyx will keep a little protein inside, and whenever an immune cells will get close to it, it will be presented to that T cells or whatever cells.
The glycocalyx acted like a sieve that basically it's a structure of glycoprotein that is above the endothelial cells. And then the lymphatic endothelial cells, they also constantly present antigen to the T cell. They are not professional antigen presenting cells. They are not like dendritic cells. So they present these little pieces of protein, this peptide to the T cell. People are starting in the last few years really to recognize the immunogenic role of lymphatic endothelial cells.
So we have a pretty thick glycocalyx here. It's a bit thicker than the wall. And that's probably because of that role.
It's very thick, also the one in blood capillaries. And I think it has two roles, probably the glycocalyx. One, definitely the rolling of immune cells. It facilitates, you know, the immune cells circulation into the vessel. And the other one, it really acts as a molecular sieve. So protein or molecule of different molecular size, they may exit and go into the interstitial fluid using pores or do like through the endothelial cells. So it's a role as a sieve. So these are the two major roles, facilitating the rolling of the lymphocyte, and the other one acting as a sieve for protein or molecule of different molecular size. Some will go through the pore or through a space which is between two endothelial cells. And some very small one, they can even enter the cells and exit on the other side.
Right.
People have shown that lymphatic are very important in different pathologies, not only autoimmune disease, in cancer, for example. There is David Leiden at Cornell that is studying, for example, the role of exosomes. And the exosome, there are these intracellular vesicles, which are released by different cells. And when they are released by cancer cells, they basically help seeding an organ for future metastasis. They facilitate when the cancer cell will arrive, this extracellular vesicle already primed the tissue for the metastasis.
So let's remind, exosomes, they are inside those big vesicule, the multivesicular body, they call that MVB.
Yes,
they have a vesicle with a bunch of exosomes that are going to be released extracellular.
Yes.
They are very small, the exosome 30 to 250 nanometer, I remember that, but it's everywhere, right? You find in saliva, blood, urine, several.
Yes, yeah.
And so you said some cancers are carried by those exosome to
they release, like you have a primary tumor, they start releasing the exosome that they go to a different organ and through the lymphatic or through the blood, and they help priming the organ for when the metastatic, you know, the cells from the primary tubule will arrive there. And so it's a facilitating factor.
So you can have protein, you can have lipid, you can have a lot of genetic material.
Yes, yeah. They've been characterized very well at the proteomic level, all the protein that they have. Yes.
RNA. So all that is carried through. Now the lymph carry a lot of exosome. You have a lot of them in the lymph.
Yes, there are a lot of exosomes. Yes, in this paper that again I mentioned that came out 2 weeks ago, we also and actually the lab of David Leiden at Cornell analyzed the exosome for us, and there is a very good amount of exosome. Yeah.
So is there practical application for diagnostic to find what type of exosome?
Yes, people are very interested diagnostically and therapeutically. Because, you know, you can use them if you can package whatever you want into the exosome, they can be used therapeutically, and there are, you know, groups that are doing that, and diagnostically, yes, yeah. So
let's come back to aging. So we block the lymphatic of the brain. We have some kind of early aging. We saw the lymphatics have some kind of hyperpermeability, and the pathogen escape from the lymph collectors when with aging. So what has happened with aging for the lymph system and aging of the immune system, immunosenescence in general? Can you talk about that? You know about more about this?
Well, in the lymphatic system, we know that a lot of anatomical changes are going on. For example, the pumping capacity diminish, and that's why, you know, if you see young lymphatic versus, you know, lymphatic from an older person, they pump less efficiently. And the amplitude, the frequency is decreased. So if you look at the lymph, it's more sluggish instead of being really transported, you know, towards the lymph node. Sometimes you see almost it's like goes back and forth, like you have some stasis. And also the glycocalyx become thinner, and we measure by ultra microscopy. And that's why, you know, they are less efficient in transporting the pathogen to the lymph node. And altogether, you have immune aging. I mean, one of the major issues is obviously the thymus. You don't have these new waves of T cells that normally comes out from the thymus. And so you rely on the T cells that you have in your body. And so many of them, whereas younger people, they have like a broader T cell repertoire, it starts becoming more and more focused with aging. And as you know, old people do not respond so well to vaccination. They
don't have a
both the T cells and the B cell counterpart, they don't make enough antibody. The titers are not very high, and sometimes the antibody don't last as long as you know they should, or you know, in a younger person. And the same, the T cells, they do not proliferate enough to whatever immunogen you are giving, and that's all together. It's called immunosenescence.
Right. And also you published something about the lymphatic dysfunction with smoking.
Yeah, that was the trouble. We were actually, it's not from our group, it's Hasina Oz-Fried at Cornell. And we were helping her on that project. But she was analyzing how chronic smoking really compromise not only, as we all know, the alveolar cells in the lung, but also the lymphatic. There is an inflammation in the lymphatic, there is thrombosis. Even though the coagulation system, all the components of the coagulation system are more prominent in the blood, but you also have several of these proteins which are part of the coagulation cascade that you also have in the lymph. And so she can see thrombosis of the lymphatic system during chronic smoking. She can see inflammation, and overall, they do not perform as well as the lymphatic from, in this case, where mice which do not smoke. They have these special chambers in which the mice basically smoke every day for a certain amount of time, similar to a chronic smoker. And again, the lymphatic in the lung are obviously very important because, as you know, we inhale constantly, air which is not sterile. And so there are a lot of particles that they need to be removed. And through the activity of, again, the macrophages, which are present in the lung, but also the lymphatic system that transport all of this to the mediastinical lymph node. And all this system does not work and does not function as well in mice that are chronically exposed to smoke.
So this thrombosis or clotting of the lymphatic precede emphysema in those smokers.
Right? Part is also the emphysema is a lot due with the destruction of the all the extracellular matrix, as well as the cells which line the alveola in the lungs, and it's just really due to this chronic inflammation that is present in these people. I mean, many of the products which are in the smokes are toxic, and so they create damage to the cells, and they create constant activation of the immune system. That, when the immune system is activated, like granulocyte, they release a lot of enzyme. Some of these enzymes kill pathogen, but they are also killing. They are also destroying the extracellular matrix, and that contributes to the emphysema, together with the dysfunctionality of the lymphatic system.
And when you blocked the lymphatic drainage of the brain, what change did you see? We talk about the redox changes, and but
we see change. Helena see in rats at least at the beginning an increase in the pressure in the brain. What we see, we measure in the cerebrospinal fluid. We see IL-1, which is a pro-inflammatory cytokine. And we also see an increase in so-called DAMPs, which are damage-associated molecular patterns. Every time the cells is damaged, it releases certain protein, which is a signature of damage. And normally you don't see extracellularly unless the cells is damaged, because these are proteins that normally are intracellular. And so we see an increase of so-called DAMPs in the cerebrospinal fluid. And when we look at area like the hippocampus, we start seeing a lot of signs of oxidative stress, particularly at the end of neurons. The mitochondria that seems to be the one more affected are a particular subtype of mitochondria, which are the ones that are present at the neuronal end of the axonal body. And these are very early signs. They are metabolic changes. When we look at these mice and rats early on in the first few months when you close the lymphatic. And so the major signature is really like a metabolic signature. It doesn't translate yet in tissue damage. And probably at that point, if you can restore the equilibrium, you may still go back to a physiological system. If you persist in that, the metabolic signature eventually will become, the biochemical metabolic signature will become in more micro damage and then eventually macro damage.
So a lot of low-grade inflammation, and things like that before you get anatomical damage. I know you work also a lot of autophagy. Is there anything new in the field of autophagy?
Yeah, autophagic, obviously, it's a basic mechanism. It's like a way to clean the cells, right? To transport. It's similar to the lymph in some respect, but it's intracellular. You transport all these waste product to the endosome, lysosome to be disposed. We didn't work on lymphatic endothelial cells, though. We work on dendritic cells, other immune cells, but not on lymphatic endothelial cells, which I'm sure it's also very important. Autophagy, it's important in every cell, so for sure also in lymphatic endothelial cells. But to my knowledge, there is not a lot of work done on that part. So when you have all the protein or all the organelle, for example, the mitochondria, they create a lot of reactive oxygen species, which are damaging during the process of formation of ATP. So at one point, if the organelle is too old, it needs to be disposed. And the way they dispose, you basically form a double membrane around it, and you form like a vacuole, and then you fuse together with a lysosome or an endosome to dispose the organelle. And the same, all the protein which are oxidized, they are misfolded. Through autophagy, they are disposed through the lysosome.
What's your area of study right now? Any new exciting?
Right now, we are definitely very interested in the lymph, and we are working on this microbiome. So here you see that we are cannulating a lymphatics. This is a mesenteric lymphatic. See, the cannula is around 10, 15 micron, and you can see the lymph. See, it basically uses the pumping activity of the lymphatic for the lymph to enter in the pipette. And so it's a complicated process to do, but it's the only way. If you want to study the microbiome that goes to the lymph node, to the mesenteric node, you need to collect the...
So that's mesenteric vessel in a rat?
That's no, that's a mouse, yeah.
And that's after a meal, right? Because it's so white. You give them a...
Yes, that's to show you, that's the part of the gut that we are analyzing. This is the colon. And you can see the lymphatic, normally they are covered by fat. And so you need to dissect out all the fat to see the lymphatics, and that's what we are cannulating. And these are the lymphatic that goes to the deep or the cervical lymph node. So this is a lymphatic that is entering, and you can see we are ligating the lymphatic in this. And we can also do in people. This was one of the specimens that we were discussing before that we get from the surgeon. This is a lymphatic. They are much bigger, so it's easy. Here we just injected the blue just to show the visualization of the lymphatics, and here you can see the cannula, so we can...
That's human, yeah, that's human. You put a diet with visualization.
That's human, yes, yeah.
Any hypothesis why we don't have lymphatics in the brain, in the placenta, in the brain parenchyma?
Yeah, that's right. We have it in the bone marrow, so we don't. And the spleen have efferent but not afferent lymphatics. I don't know. I mean, it is in the meninges, and people are still debating exactly, how much waste product goes to the CSF, into the lymphatic, and also, as you know, in the brain there are present the glymphatic, which are these vascular spaces. There are a lot of groups. It's a very hot area right now.
It would be nice to know how much immune surveillance we have in the lymphatics, how much lymphocyte, macrophage, dendritic cell.
Well, the meninges is definitely a very hot area for immunosurveillance.
And it was discovered by Italian anatomist Paolo Mascagni, 1837 or something like that. So,
right.
So it's not it's a rediscovery really of this.
Yeah, people forgot for a long time, and then they were rediscovered, right? And the same in the spleen, you don't have afferent lymphatic, but you have efferent. So, they get out from the spleen, but they don't enter. In the bone marrow, the same, you don't have afferent lymphatics.
You say that is, they're afraid in the placenta, in the brain, they have too much immune reaction. Is that logical?
Possible. It's possible that, because they are both organs in which you do not have an immune response. And so maybe they were considered like long ago, immunoprivileged sites, like the immune system would not enter. Now we know that it's not completely true, like immune cells can enter, but definitely they are still privileged as compared to other organs.
Some immune cells are present in the very low concentration brain.
Yeah, I mean, Beckerle many years ago, like 30 years ago, showed that the T cell can patrol also the brain. They can enter through the meninges and then they exit. There are a lot of dendritic cells in the meninges, so they are outside the brain, but they are, again, it's a sort of patrolling, because through the lymphatic, they have a notion of what's going on in the brain. They receive the lymph, and we know that the lymph that enters the lymphatic partially, in part, are derived from the CSF. And,
so in the CSF, do we find lymphocyte and macrophage dendritic? We find all those?
Normally, there are not immune cells under physiological condition, no. Only when the blood-brain barrier is compromised, at that point, yes, yeah.
They say now they almost find a microbiome in the brain, a specific area of microbiome. So we might have a lot of virus, bacteria, and so they just...
yes, yeah, they found it in many... With the sensitivity of many of these techniques, from proteomic to genomic, is definitely increasing. So people start seeing things that before it was not possible to see. Also in tumors, like people find a particular microbiome that is associated with tumors. It's still very open question, what is the role of it? Like if it would help, or if it's conducive, or if it's protective in terms of cancer immunity.
And in the bone marrow, you basically have so much immune cells that will explain why you don't really need specific vessels lymphatic vessels for it.
It's possible, or also maybe, as you say, I mean, they are privileged site. You don't want to have an immune response. I mean, there are immune responses actually in the bone marrow, but I don't know. I think like the placenta, for sure, you don't want to have an immune response there.
In the interstitium, the extracellular fluid, you have very, very high concentration of antigen there, even more than the lymph. But so in the of the brain, we don't have that.
No, you also we find a lot of antigen protein specific in the lymphatic that are draining from the meninge, and they arrive to the deep cervical node. We have a so-called tissue-specific antigen. Yes, yes, they are like an enriched protein that are, particularly enriched in a certain organ as compared to another, like MBP, PLP, this protein which are associated with the brain proteome.
That's fantastic. That's so interesting. All right. Thank you so much, Laura. It was so interesting. I don't want to tire you too much. You have to work today.
My pleasure. Thank you so much for having me.